So thank you, Sarah. First of all, I'd really like to thank the organisers. I think I probably speak a little bit on behalf of everyone when I say I'm absolutely blown away and amazed how much bigger this foundation has got in a year. I think it's absolutely incredible, and it really speaks volumes to all the parents that have contributed their time. As I said to someone last night, if only university admin was as efficient as you guys, we'd be a much better place. So I recognise a few familiar faces from last year. Those that I don't or have forgotten who I am, I'm Dr. Nathan Absalom from Western Sydney University. Mary and Ricky did a really great job of explaining the large collaborative group that I work with. So genotype phenotype correlations, what are we doing and why? With a genotype phenotype correlation, what we're doing is we're looking at how GABA-A receptor variants cause epilepsy, so that's the first question. And the second, what are the consequences of different changes to that receptor that have been caused by this variant. And you would have heard a lot about loss and gain of function today, and that's the main one. But let's just take a little bit of a step back and think about, well, what can go wrong with these receptors in the first place? And the first thing is you can have variants that either introduce a deletion or maybe affect trafficking, and we call those basically LOF null, and it stands to reason, right? If you have receptors and a variant stops them getting to the cell surface and they're not there, you're not going to get as much chloride go through the receptor, and epilepsy will follow. That's uncontroversial. Another thing that I would say is uncontroversial, and again we've known it for 20 years, is that if you have receptors that do get to the cell surface, but they change one of these gating equilibrium constants, and they stop the receptor from opening. So for instance, it might be a variant at a GABA binding site, and GABA struggles to bind at the same affinity at that binding site. You can measure a concentration response curve that shifts to the right, so you need more GABA to open the receptor. That's called a loss of function in the red. And again, stands to reason, you'll get less chloride coming through, and you'll end up with epilepsy. Again, very uncontroversial. Third simple model is, okay, the receptors now get to the cell surface, but they change these equilibrium constants in a different way, so they make the cell more open. They're more likely to be open, so they open in response to lower GABA concentration, so here the blue, the gain of function, or they just open for the hell of it, and they're just constitutively active. So if this happens, it stands to reason that the chloride currents, I put inhibitory currents there, that's probably wrong, stands to reason that chloride currents are increased, and it's actually a little bit difficult for us to predict the clinical outcomes that follow. And I think that's been a real problem for the field. So, how do we know what that means for the patients? What are the clinical outcomes? And this is where we do a genotype phenotype correlation. And this is not something anybody can do alone. And this is why we have to collaborate with such wonderful people. Ricky's not in the room. Damn it. So we collaborate with people from Denmark, so Ricky and Sebastian, and as you know, they do interviews with patients and clinicians, have been really useful, this foundation, in getting that data. And they collect all that data, and then they tell us the variant identity. This is a scientific group. It also includes Chris's group that work with mice and us that work on the in vitro work. And we determine whether it's a loss or a gain of function, and then feed back that information to Ricky and Sebastian, and then we sit down and somebody takes responsibility for analysing all that data, which is actually probably the longest part of this process. Just saying thank you to the funding partners up there. So what do we find out when we do this? So Mary introduced the two projects I'm going to talk about, one on the GABRB2 gene and one on the GABRG2 gene. So we'll start with this. And here we compared loss of function missense with gain of function. So what we had is we deliberately concentrated on variants in the transmembrane region, and we got high quality data for all these variants in terms of the clinical data. And what we did is we measured concentration response curves first to determine if they are loss or gain of function. To do that, I'll just spend a second explaining this. So what we do is we create a single strand of DNA which has all the 5 subunits concatenated and joined together. There's a couple of reasons we do this. The first is so that we're measuring one specific receptor. So we have less variance. It's technically very useful for that. It's also so that we can introduce just a single missense variant into the receptor construct. And that means it represents the heterozygous state. That's quite important because all the children have only one copy of the receptor and one copy of the wild type. unless you're alpha-3 because it's X-linked, but I'm not going to go into that because that's just too complicated for right now. It took us about 2 years to perfect this system and to design all the controls. That's usually a death sentence in academia, but lucky I've got a very good boss that didn't sack me. And what we do is we inject that into xenopus and we record the currents that are listed by different concentrations of GABA. And here in black we've got the wild type, and we measure the concentration of GABA that elicits 50% response. So you can see with the blue you get larger responses, and that's a gain of function, and in the red you get lower responses, and that's a loss of function. So what we did from there is we then compared the loss of function to the gain of function phenotypes. And what we have here is the different phenotypes that are more likely to be found in the loss in the blue dot, more likely to be found in the gain in the blue dots. So severe intellectual disability, microcephaly, movement disorders, feeding difficulties. We also have increased early mortality with the gain of functions. Fever sensitivity, so febrile seizures, are more prevalent in the loss group. That's what really distinguishes the two groups. And we actually didn't see any difference between seizure freedom and hypertonia. Now, that is all fairly similar to what we've already published with the beta-3, but what was different was this age of onset. So you can see the loss of function, and they're pretty much the same as any of the receptors, between 6 and 24 months, and nice and tight distribution. But there's this really big distribution with the gain of functions. And so we say they're not different because the mean or the median's not different, but the distribution's really different. So they're actually quite different, these two data sets. And if you look at seizure risk, again, you don't see any significant difference when you measure it by that. So we were a bit intrigued. What could be causing this difference? So what we then did, and we looked at our data and we thought about it, we said, well, hang on, these aren't all going to be equal. Some variants have different magnitudes of change in EC50. So some shift the curve a little bit to the left and some shifted a long way to the left, and some introduce constitutive activity. So what we did is we binned our data. Unfortunately, the colours aren't so good there. It was a nice data set to do that because it was 2 cohorts. And when we did that, we found with the phenotype associations, there wasn't too much difference except for feeding difficulties. I wasn't going to say anything about this, but I'm going to stop there for just half a second because of Martina's lovely presentations. I usually see that as kind of a dot and haven't really thought about the consequences of it. So I'd just like to thank you for bringing that, and I'd like to thank all the patients' families that we've talked to today for bringing these sorts of things to our attention and challenging us to be better scientists. And I really mean that. I really thank you for that. So I'll just move on back to the science now. We saw some difference with the survival, but it wasn't significant because the groups weren't large enough. And when we looked at the seizure onset, the age of seizure onset, what we saw is that we got this nice differential between the high sensitivity gain of functions, where we saw seizures at a very young age, and then with the low shift, we saw some with no seizures at all and some with seizures at a much later age of onset. So we can see that, I mean, some of you have been saying, I don't get seizures. Or I got seizures very late, and that's probably due to the magnitude of change of your variant. And so that's really nice because we hadn't got that sort of data before. And you can see the seizure risk is very significantly changed. Oh, there's a nice little graph there. So the take home from this study was that we have basically 3 distinct clinical phenotypes. We have the loss of function missense. They're a different syndrome. We often say that they're less severe, but that's comparatively. I don't want to make it a competition between loss and gain. It's a different syndrome with different challenges. I think that's the best way to describe it. So you're on the GEFS spectrum, have an age of onset of greater than 3 months, and you often have mild to severe developmental delay or different comorbidities. With the gain of function, it sort of separates into 2 groups, but really think of it more as a spectrum. Yep, going from low to high, and the higher that your change in the EC50 is, the younger your age of onset, the more likely you are to have these severe epilepsies like IMFS and IESS, and in both cases you're likely to be non-verbal or non-ambulant. You've got high rates of that. So that's great, but they weren't all the groups that I showed you at the start. And so at the start, I showed you there are these loss of function null and loss of function missense variants. And for other genes, these types of variants actually have quite different outcomes. So if you look at SCN1A variants, for instance, loss of function variants that are missense and change the channel activity have less severe outcomes than null variants that get to the cell surface. So if you don't get to the cell surface, the phenotype's worse. But is that the case in GABA receptors? Nobody really looked. So we did. And we did this in the gamma-2 because we got a lot more variants in the gamma-2 that were null or null frame shift or basically deletion variants. So we took all those variants and we compared them to a set of variants and we measured them and showed they were loss of function missense. I won't go into that too much. And we compared the clinical phenotypes between the two. And here's what we get. We found that the epilepsy phenotypes, so we've got null there and the missense slough, we can see that a lot of the nulls don't actually develop epilepsy, they just have febrile seizures. And some of them just have febrile seizures plus, whereas missense sloughs much more likely to have DEEs. The age of onset isn't particularly different, it's about the same. But when we look at the comorbidities and some of the aspects of it, so for instance, inheritance, this is a big one. If you have a null variant, you're much more likely to have children. Gain of function variants, I don't think we have. We do in the alpha-3, again, that's different because it's X-linked, but apart from that, we don't see any inheritant variants or de novo. You have increased seizure frequency if you have a missense, increased levels of intellectual disability, language development, psychiatries, motor coordination delay, and hypertonia. So in all these, what we see is that loss of function missense variants are more severe than null. And whoa, we can actually, so we did our odds ratio test, but what we found here is because these are the same, we can estimate the severity just by doing a simple cumulative severity score where we add up if it has de novo, if it's de novo, if it has intellectual disability or language delay, we can score it 1 or 2 or whatever. And when we compare the cumulative scores of the loss of function versus the null, we see highly significant difference in the severity of those disorders. So what we can see is that the loss of function nulls are quantitatively different as a group to the loss of function missense. So what this means is now, as we're doing more and more of this work, we find that we can categorise your variants into much more discrete categories. So we have loss of function null, that should say less severe. I must have copied and pasted. And loss of function missense is more severe, but they're basically two different spectrums of the same syndrome. And then gain of function is its own syndrome entirely. And again, it has a spectrum that goes from low shift, low changes in the EC50 to high shift, and has those different aspects to it. So very, very quickly, there's just a little bit to think about with treatment strategies. If you think about, I hadn't thought about Paul's work very much, but I was thinking more about the SCN1A gene therapy, which appears to be successful going to clinical trials where they target the poison exons and they just drive as much sodium channel up to the receptor regardless of whether it's good or not. And we should just be a little bit careful if we're pursuing a strategy like that, because any strategy that gets more of defective receptors to the cell surface is probably not such a good idea. When it comes to ASOs, really big issue is, well, what happens if you take down all the receptors and you go too far the other way? I think we can be a little bit more positive on that because looking at the nulls, it suggests that the therapeutic window is probably a bit larger than it originally is, with the caveat that I've only looked at heterozygous null variants, not homozygous null variants. So there's going to be a floor that we can't pass, that we don't want to get it below, but that floor is probably a little bit, a few floors below what we expected, and so that's good. So I just wanted to bring that up. And I think that's it. Yeah. Thank you.